Hi, I would like to discuss about a topic called clinical research now. You know, clinical research is known to everyone that human volunteers are taken for drug trials. They would be taken for different drugs and checked for any clinical safety. The same, it's exactly the same, but there is something you should know more about clinical research. It's not just making the trials on the human subjects. When the clinical research has to be done, it should be coming from after the preclinical research. Preclinical research is done on the animals or any other in vitro systems, and after this, the clinical research is done. And this clinical research is after this is done, it comes into the market, and finally we take the medicines. So, so many people would oppose this clinical trials that human volunteers are taken for studies and they would be harassed. These kind of stories we are hearing in the news. But understanding the principles and the key concepts of clinical trials would generally people would appreciate the clinical trials. This is the safety procedure done before the drug comes into the market. So I would like to discuss about the clinical research, these clinical trials in very detail now. Just I would like to explain about the randomized clinical trials now. So this is clinical research is done only to evaluate the new drug or procedure or any device on human volunteers to check for any clinical safety. So this is the main aim of the clinical trial. And talk about the sponsors. And the sponsors would be pharmaceutical companies or biotechnology companies or some of the charitable agents. These would sponsor the process. And when the clinical research is done, the patients and the clinical investigators, the trial personnel which are going to do the trial, must not be known about the topic or the any program what they are going to do, because if it is known to them, it may go to the biased result. So both of them must not be aware of the what trial are going to be conducted. But there should be some ethical manner in conducting these clinical trials, such that the volunteer should not get compromised with their personal satisfaction. They should be treated in a very kind way, and they should be voluntarily taken the informed consent. After this is all done, the clinical trial is done on them. So there are many types of trials, and the clinical investigation is done by clinical investigators, mainly of three main rules. To look at the best way to give or withdraw drugs, and second point is to investigate the best duration of treatment to maximize the outcome, and the third point is to assess the benefits of prevention with vaccination or screening programs. So these are the three main concepts of clinical investigators. And this clinical research is done in 4 phases. We categorize them into 4 phases. Like phase 1, in which the pharmacokinetic and pharmacodynamic parameters are mostly considered, and this is for immediate short-term clinical safety data. And in this phase, only the healthy subjects are taken, with very few numbers. And in the phase 2, in this we can take specific disease persons or healthy volunteers. In this, the dose-response relationship is calculated. This area is mostly concentrated. The dose-response relationship is mostly concentrated. And in this phase 3, this is a very critical step in the clinical research, because this is the stage where the drug gets access, whether it will get marketed or not. So when the drug has been approved, it is called as landmark. And this is the phase where very large population is taken, a wide range of population will be taken, and where the people of specific diseases are taken, not just healthy human volunteers. Specific disease persons are taken and they are done for clinical research. Okay, the drug has been marketed now. So this comes into the 4th phase. This is called the phase 4. This is called as post-marketing surveillance. Even though the drug has been marketed into the marketed it has reached the people. All the medication is taken by the doctors, but this is done in a very wide range population, and to assess the safety after the phase 3, to see if there are any adverse effects or any kind of side effects, only to assess the clinical safety, these phases are done. And these are all about the phases. Now we shall discuss about the trial design. So these are all about the phases, and now the trial design is of many types. First one is the parallel trials. Second one is crossover trials. Third one is factorial trials. Fourth one is clustered randomized trials. So all four trials have different specifications. So let's discuss about the first one is the parallel trials. In the parallel trials, the subjects are taken, and these are taken, every subject are taken at least either for one group. Like if they are taken for test group, they are only treated with the test group. And if another person is treated for this control group, like a placebo study, they are only for the study. They are not later taken for this study. They are not changed from themselves. So they are studied in a parallel way. So this is the parallel design. And what is this crossover trials? Crossover trials in which the healthy volunteers are taken, but at some time, even though the person has been taken for the trial group, but sometime after the trial is done, he would be taken for the standard also. Likewise, if the person has taken for the standard, he would also be taken for the trial. So every subject is going into all the phases, all the groups. So this is called crossover trials. And the third one is factorial trials. In the factorial trials, the volunteers are randomized for the test group A. For example, take the test group A, and these are compared with the placebo study. And again, these persons are taken for another group like trial B, and we randomize it there and compared to the placebo group. So this is called as factorial trial. And the fourth one is cluster randomized trial. In the cluster randomized trial, in which very large population is taken, especially by a single practitioner or hospital, and done on the patient, on the volunteers. These are the different types of trials designed, and these trials are done at different places. So there are two types of centers: single center and multi-center. Single center studies are mostly done in the phase 1 and phase 2 trials, and in the multi-center studies, these are done at various places all over the world sometimes. This is mainly done in the phase 3 critical step. And there is some reason behind why multi-center studies are mostly preferred, because to evaluate a new medication or procedure more efficiently in terms of accruing sufficient subjects over a shorter period of time, to provide a better basis for the subsequent generalization of the trial's findings, that is, the effects of the treatment are evaluated in many types of centers. So this is the reason why the multi-center studies are taken. And there are other classifications of clinical trials like superiority study. Superiority study is done when the new drug has been taken into the clinical research, and where we are going to show that the new drug is having more efficacy than the present drug which we have. So those type of studies are called as superiority studies. And the second one is the equivalence study. The equivalence study is taken by comparing with the standard one. We want to show that this new drug which we are going to make the trials is going to meet the specific minimum requirements of the standard. It's not equally taken into the standard one. It may show this much of effect, but this is efficient. So those are called equivalence studies. And the third one is non-inferiority study. In this non-inferiority study, you take a comparison with the standard, and the standard is having this much of efficacy, and you cannot just ignore that this new drug is also having less efficacy, but it has almost equivalent to this standard one. So this is called non-inferiority study. And there are two types of studies, like exploratory and confirmatory. Exploratory, the word itself indicates that it is going to find out something new. Those type of studies are called exploratory studies. And in the confirmatory studies, you take a subset of some study and you're going to find out what is inside. You want to explore it more detail, like to confirm what is present there. So that is called as a confirmatory study. And even though the clinical trials are done by all these places and these are all parameters taken, but we still find some of the errors. There are many types of errors called bias or systematic errors, confounding errors, or random or chance errors. And these errors are mostly seen after the drug has been marketed out and affecting the population. So this may be due to some of the bias or systematic errors. I will explain about the bias systematic errors now. These bias systematic errors are, for example, if you take, if the clinical investigator is known about what type of drug it is and what type of treatment he is going to look at for. If he is known about it, and he may look for the specific outcome, what he wants, and how many volunteers should be taken into the study, that type of study, he would take with his own ideas that would hide some of the factors that would lead to some of the errors. So the clinical investigator also must not be known what type of study he is doing, so that he would take in a random way. And the second one is the confounding error. Confounding error is seen when, for example, the person is taken for we are taking two groups, test one and the standard one. And if smokers are taken for this test one group, and non-smokers, only non-smokers into one group, and look at for the study and test A, test B, and you can perfectly say that obviously even non-smokers would be having greater efficacy. That is, you cannot just simply say that non-smokers have greater efficacy. There's something missing in that. This is called confounding. So these type of errors are called confounding factors. And so to remove these confounding factors, we have to know what type of factors which we want to do. We have to, before doing the experiment, before doing the trial, we have to know what factors might come in the trial, what type of confounding factors may come. So based on that, we have to do this stratified randomization. And after the clinical research is going on, to statistically find out and to reduce the significant confounding factors, we use regression analysis. So likewise, we can reduce the errors. The third one we explain about random errors. Random errors are mostly seen because of biological variability. This is not stopped at all. The previous two factors can be stopped, but this is obviously it is very rare event which we can see, and this is because of biological unsimilarity. So the person, this is about the random error, and even though the drug has been marketed and it shows some adverse effects, it is due to random error or chance error called as. And if the p-value is less than 0.05, we take a probability factor now in the statistical systems. If it is less than 0.05, we can say that the result which we got is because not just of random error, it is statistically called significant. And if we got the result greater than 0.05, then we can say that this is due to some of the random errors, and it all is not statistically significant. These are all about the errors which we can see in the clinical trials. So this is all about the clinical trials, and in the clinical trials, the children are not taken into the study, and also the pregnants are also not taken. This is all about the clinical research. Thank you.